She attended a workshop by you at Shulini and was very impressed. So she says hello to you.
Hello, hello, how are you?
So we are live on YouTube, and I think it's time to start. What a wonderful day. It's starting to rain here. It is thundering. Narendra, I don't know. Before we start, actually, before we get into COVID and Corona, let us test your skills on geography. But
Oh,
clearly the temperature is, 3 to 4 degrees lower here in India, at least in Solon. It used to start getting warm, but now it doesn't get warm. Is it because of pollution, you think? Is it the same in the US or it's different there?
well, actually, in Philadelphia and New Jersey, it's still pretty cold. It's 1 or 2 degrees in the night most of the time, and I actually don't go out, so I really don't know what the weather is. I'm doing my social duty of social distancing, but I think I see news from all over the world. Things are getting so much more prettier. The nature is surely affected.
Wonderful. So, once again, welcome to the first episode of Expert Talks by Shulini University. And who could be better to inaugurate that than Dr. Narendra Chirmule? Narendra is, as all of you know, a visiting professor with Shulini University, but more importantly, he's a fascinating speaker. He's the CEO of SymphonyTech Biologics, which is using analytics and biology together. Narendra has been the ex-R&D head of Biocon and also been with Amgen, as which all of you know is the world's leading biotechnology player. He's part of the COVID-19 workforce for the government. And who could be better to talk to us about Corona than Narendra Chirmule? But before we actually get into this very exciting topic, let's just probe a few questions with Narendra. What's really happened? Where did this coronavirus come from, Naren? And why would you think it's so much more prevalent in the US versus India? Has India done much better, or is our genes better? Is the weather supporting India, or were we able to lock down at a better time? What are your thoughts about these issues?
Yeah, so the first question about the source of the virus, I think there is enough evidence now coming out of the literature from the sequence analysis that it seems to be very close relative to the virus, the virus that infects the bat. So I think sequence analysis will tell us ultimately the source of this virus. As far as why India has a much lower infection rate rather than the rest of the world, I think there are multiple reasons. No one reason. I think there are a combination of many things. One, surely the lockdown played a big role. So the earlier we had the earlier you do the lockdown, the better it is. The other thing that is very interesting in India is that this is almost the third time in India we all have come around a single issue. For example, we were together with demonetization. We were together with CAA. You could have been on either side of the coin in terms of discussion, but you were definitely on that one topic. And this is the third time we are coming together as a nation to discuss. The whole nation is talking about one topic. So while the rest of the world is trying to figure out how to talk together, India has done it 3 times. So we are sort of used to working together on a single topic. So I think that is a single most advantage I feel India has had socially. But then scientifically, I think there are a lot of data that will come out in the future, I guess, about the hygiene hypothesis, whether exposure to different pathogens makes you a better immunity to this virus. I'll speak a little bit about that in this talk. So many reasons, not one reason. So I think time will tell. It's literally history in the making.
So I won't come in between your talk and the audience. So let's start, Naren. Over to you.
Great. Again, thank you so much, Atul and the Shulini team for making this happen. I am inspired by the work you do, Atul. And every time I talk to you, there's something new up your sleeve. So thank you again. So let me share my deck, and I'm going to minimize this a little bit. So I'm going to talk about my thoughts about what it takes to develop a vaccine against this virus. my experience in vaccines has been varied. So I'm going to share some of my experiences and how this vaccine will be developed, and all the positive and the negative things that we've learned over the last several decades. So the topics I'll cover are these, which is basic immunology, the components of the virus, the immune response to the virus, a process of vaccine development, a rational design. What does it take to design such a virus, such a vaccine? And then we touch a little bit upon herd immunity. Since the audience may be very diverse, let's talk a little bit about basic immunology because it's relevant to understanding how a vaccine works. So I like the immunology of war to study basic immunology. So when an invader invades, the invader comes with full force. The border police initially checks this invasion. But if the border police is overwhelmed, then it is time for the army to come. And when the army comes, you need a general who sort of coordinates the entire war and instructs the Navy and the Air Force and the army to do their thing to come and attack the invader. The army and the Navy very efficiently come, attack the invader through their own mechanisms, and then finally eliminate the invader. Not very different from what happens in the immune system. The virus enters the body, and there are lots of mechanisms initially to try to block the virus from entering. However, the virus overwhelms the immune system and then enters the body, no doubt. The immune system is equipped in transmitting the message that the invader has come to a special cell in the body called the CD4 positive T cell, which in the analogy is your general. The CD4 positive cell instructs B cells to make antibodies, which can then bind to the virus and clear the virus. And this is the analogy is the B cells are primarily in lymph nodes, and they secrete antibodies, which like missiles go all through the body. And they also activate CD4 cells, also activate cytotoxic T cells, which are cytocell toxic, which they are able to kill the virally infected cells, almost like hand-to-hand combat. So therefore, it's the analogy of the army. The cell-mediated immunity is mediated primarily by cytotoxic T cells. The humoral immunity is mediated by neutralizing antibodies. And a vaccine has to elicit these responses. But before we go into the depth of the vaccine, what I'd like to do is just explain to you what cell-mediated immunity is, because it's relevant to developing a vaccine. When an intracellular antigen, which is like the virus inside the cell, needs to be recognized by the immune system, it is presented by a particular pathway and presented by a molecule called MHC class 1. And this molecule is then recognized by CD8 T cells. The presentation of the virus peptide activates these CD8 cytotoxic T cells. These T cells kill virally infected cell, and this comprises of the cell-mediated immunity. On the other hand, external antigens or extracellular antigens are like the virus enters the cell, and there's a different pathway that the antigens are presented to another cell type, another MHC molecule called MHC class 2. These present antigen to CD4 cells. Presentation of the virus peptide by MHC class 2 activates CD4 cells, and CD4 cells help B cells make antibodies, and this is called humoral immune responses. And so the immune response is highly regulated, and the reason it's regulated is because it is a balance between responding to an invader or virus versus not responding to something that is self. John Wiley was one of the pioneers of understanding the structure of these MHC molecules and how it presents antigen. And he was one of the most influential biologists of his generation for his discoveries on how these immune systems protects humans from infection. So now, what are the factors that allow your immune system to recognize the virus and kill it? All of us respond to the same pathogen in different ways, and we know this because from all viruses, and including this one, some people are exposed to the virus, have no symptoms. Other people have very strong symptoms and die. And there is a whole range of reactivity in between. So why is there such a differential response to the virus in so many people? Because a lot of factors are important in regulating the immune system. And I just list a few. The first is the affinity of the virus to the immune cells is different in different individuals. The extent of the initial inflammatory response that is induced by this interaction, the genetic factors of the individual, which is the MHC type. We all inherit our MHC from our parents. So we only have our ability to respond to the virus is dependent on our genetic sequence. And then the affinity of this MHC peptide to bind to T cells. And then finally, the number of immune cells that are activated during the course of this recognition process. All of these combined are what is used for designing the optimal vaccine for this virus. So let's talk a little bit about the virus. It'll be a recap. I know a lot of people are reading a lot of information. The virus is made up of structural proteins. It has an RNA nucleotide. The structural proteins are the spike protein, envelope, hemagglutinin esterase, membrane, and nucleocapsid proteins. A lot of focus on the vaccine has been on the spike protein, which is responsible for entry of the virus into the cell. Let's look at the spike protein on a structural basis. It is a molecule on the surface of the virus, which has a proteolytic cleavage site, which means all of these enzymes, which are present on the surface of the cell, have to cut this protein into two before the virus can enter the cell. And that I've shown in the next slide as a cartoon. So there are many steps involved in the virus for virus to enter the cell. It has to first bind, and then it has to cleave, and then it has to fuse. So in the cartoon, what I'd like to show is this red and purple dot that I've shown here is the spike proteins S1 and S2 regions. So the virus binds to the ACE receptor. It gets proteolytically cleaved and then fuses with the membrane and infects the cell. Let me repeat that. It binds to ACE2, it is cleaved and fuses with the cell membrane and infects the cell. So it's not a simple bind and enter, it is a multiple step process. So for this, the vaccine should be able to not only block the binding of the virus to the receptor, it should also develop antibodies to block the fusion process. It's a tall order. From structural biology studies that have been done as recent as March, we know that the trimer of the spike protein on the cell surface of the virus is a required structural element for the virus to enter, and which creates a particular, what is called an epitope that binds to the ACE2 receptor and the virus enters the cell. And I just showed you in the previous slide that the S1 and S2 portion needs to be cleaved before the virus can enter. So it's a very complex mechanism of viral entry. So now let's look at the immune response to what patients have been responding and how they have been responding. All patients exposed to the virus develop antibodies. Antibodies are directed to the spike protein, and antibodies to spike proteins can block virus entry in laboratory experiments. What we're still finding out is the role of antibodies to the other proteins of the virus, like the M, E, NC, and HE. They are there, so they must be there for a reason. So we don't know what that reason is right now. What is the level of antibodies that is protective? And whether both humoral and cell-mediated immunity are required for protection. So we'll try to answer some of these questions as we design the vaccine. So as far as antibodies are concerned, there are at least 3 different kinds of antibodies which define the qualitative and quantitative nature of the immune response. First, much easier to understand are the neutralizing antibodies, where antibodies will bind to the virus, shown here in red Y-like structures. They will block the virus from binding to the receptor and therefore will block entry. There are also non-neutralizing antibodies, and these non-neutralizing antibodies will bind to the will bind structures outside the binding entry site, but they do something. We still don't know exactly what their role will be, but they will do something, but they are called non-neutralizing antibodies. And the third kind of antibodies are what are called antibody-dependent enhancing antibodies. So typically, we look at antibodies to protect us from infection. But there are also these antibodies, which are pathogenic antibodies, which can make the virus enter faster and make it worse. So the way the mechanism of that is that the antibody binds to the virus, and the non-antigen binding domain of the antibody, which is this straight line of the Y, which are called FC receptors, FC portions can bind to FC receptors on other cells and confer another mechanism of virus to infect cells. Although this mechanism is rare, it is possible to happen. So the overall course of the immune response is that after the initial infection, this virus goes into sort of an incubation period where you have asymptomatic phase where the virus is replicating. These are the people who are spreading the infection quite a bit in this epidemic. The virus load increases. You start getting symptoms, shown here in red. The immune response kicks in. You first get a primary immune response marked by IgM. Then you have T cell responses which start initiating, shown here in pink. And then finally, you get IgG secretion, which is the antibody and high concentration of antibodies. The vaccine should induce high level of antibodies and also T cells. So let's look at how you would design a vaccine for COVID. What are the questions we want to answer? Who needs to be vaccinated? What is the nature of the protective immune response? Is it just humoral antibodies? It is cytotoxic T cells? What part of the virus can induce this immune response? What kind of vaccine, what dose, what frequency is required? How much vaccine needs to be made if this is successful? And how will it be distributed? So let's start with who is to be vaccinated. And it's obvious that people who are not currently infected should be vaccinated, who are susceptible to the infection. And high on that list would be healthcare workers who are in close contact with virally infected cells. Heroes are people who are infected and getting treatment for all kinds of drugs that are available today. Then susceptible people who are older people, not able to clear the virus quickly. Those are the ones who should get vaccinated. And then healthy contacts of people. So what's called in pharmaceutical language, the pharmaceutical development language, the term for the questions to ask or the requirements is called the target product profile. So what is the target product profile? What do you want in this vaccine? A vaccine should prevent infection. The vaccine should also stop replication if the virus goes inside the body. What is the indication? It should be for healthcare workers, susceptible individuals, and healthy contacts of these people. Approximately, you will require 10 million doses, 10 million people. The measures of efficacy will be developing neutralizing antibodies and also cytotoxic T lymphocytes. The dose to induce protection based on historical results from other vaccines can be, for example, if it's a viral particle, live viral particle, it'll be around 10 to the 10th particles. If it's a protein, it'll be in the 20 to 100 microgram range. These are just averages approximately of what the dose could be. The vaccine should be durable for at least a year, and the product should be stable for 6 months to withstand shipping, staying in the doctor's office, etc. So quickly, the protective immune response, we don't know what the protective immune response is today. We assume it is going to be neutralizing antibodies. However, we also think that there should be some component of cell-mediated immunity to eliminate the viral infected cells if the virus enters the body. In terms of vaccine development life cycle, these are the high level, these are the steps of what it takes. Normally, it takes about 10 to 15 years to develop a vaccine like this. We are doing it in record time with enormous collaboration across everyone in the world. So the first step is to identify to what you're going to make the vaccine against. And a lot of studies suggest that the spike protein in this case, in this virus, seems to be the appropriate antigen against which a vaccine could be made. So in January, the paper on the structure of the spike protein was published, and in a laboratory assay, it was demonstrated that an antibody could block infection. There are many companies who are developing many vaccines. Almost 80 to 100 different companies are developing vaccines, each of them using different platforms of expressing the spike protein. A process has to be developed to manufacture the vaccine in a large scale. Animal toxicology studies need to be done, and clinical trials need to be initiated, after which there will be regulatory approval, and then finally scale up and distribution of the drug. And we hope as humanity for all of this to be done within a year and maybe less, even 2 years. So it's an astronomical task. Remarkably, all of these things have been done within months. A little technical detail about the different viral vectors, of the different vectors that are being tried for this vaccine, in their ability to either induce CD8 T cells or CD4 T cells, which means a cell-mediated immune response through CD8 or humoral immune response to CD4. So both DNA and RNA vaccines will be able to elicit both CD4 and CD8 responses. They will generally be safe, very safe. 3+ means safe. Means they don't have any major side effects, or they don't cause real inflammation at the site of the injection, etc. If you use a protein, then because it has no endogenous component, it cannot present from the intracellular path. You will only see CD4 cell activation and only antibodies, but not cytotoxic T cells. Viral vectors, which I will talk a little bit more in detail later, will induce CD4 and a strong CD8 response. Their safety profile may be slightly less than these other regimens because they do induce a little bit of inflammation. Inactivated virus, which is, for example, of the live virus, which is inactivated by either inactivation by radiation or some mechanisms, will induce the CD4 cell, but not the CD8 cells. And live attenuated viruses, which are taking the live virus, removing some gene which will make it non-pathogenic and use that as a vaccine, will be extremely efficient. However, from a safety perspective, it might have a lot of issues. The reason I'm presenting this at live attenuated virus as an option is because all the vaccines that work extremely well in humans, like measles, mumps, rubella, varicella, all the vaccines that have been approved for so many years are all live attenuated viruses, and they work extremely well. So a lot of clinical trials are underway to do these studies. I will talk a little bit about the Oxford University chimp adenovirus vector as an example of how a vaccine is developed. So the chimp adenovirus candidate vaccine, or called ChAdOx, I love the name, ChAdOx 1, is a chimpanzee adenovirus virus, which is essentially in humans, it's the adenovirus which causes the common cold. The virulent genes of the virus are removed and replaced with the gene for the spike protein of COVID. News articles have suggested that this kind of a vaccine, not for COVID, for other diseases, have been injected at least in 300 individuals. So it's a generally safe vaccine. And it has also been shown to induce both humoral antibody and cell-mediated immune responses. So in order to manufacture a vaccine for the engineering students in the audience, it's a massive process. It's a massive undertaking. You have a large manufacturing facility where you have control parameters, you have fermenters, you have purification units, and a very, very clean environment. You start from process design, you characterize the process, there are pilot lots, engineering lots, you have to validate the process, ensure that the process works systematically and dependably every time. And finally, you have commercial product and an audit of the facility. All during the entire course, there's extensive analytical testing, which ensures that you're making the right thing. Then we move to the clinical trial. How do you design a clinical trial for a vaccine? The details of this clinical trial that Oxford University are doing are presented in the Clinical Trials Gov website. The clinical goal of this vaccine is to induce protection from COVID-2 infection, and if infected, curb progression of disease. So both goals are there. And the immunological goals corresponding to them will be to induce neutralizing antibodies and also to induce cytotoxic T lymphocytes. 18 to 55-year-old subjects will be in good health will be used. A little bit more than 1,000 patients will be participating in the study. It'll be a single dose study. Volunteers will be blinded and will not know whether they have received the COVID vaccine or whether a meningococcal vaccine comparator. And after a predefined number of people are infected, the study will be unblinded. And so the steps that are involved in developing a vaccine will include phase 1, phase 2, and phase 3 studies, which will then ultimately result in developing a package of documents, which are called the biological license application, which will include many components like the pharmacology, the toxicology, the process, the analytical characterization of the molecule, all the studies from the clinical trial, all the risks will be listed, and then finally it will go into regulatory approval or regulatory review, and which will evaluate scientific efficacy and safety, and then inspection of the facility. So let's step back a little bit and consider that the vaccine trial may be done. This is just a hypothetical scenario to show what the results could be. We can predict what they can be. These are the probable possibilities. So I told you there are 1,000 subjects in the clinical trial, and maybe 100 of them get infected. And when the study is unblinded, let's say 100 people are infected, it might demonstrate that none of them got infected in the vaccine arm. All 100 got infected in the meningococcal arm. That means your efficacy of your vaccine is 100%. You could also see a result where of those 100 individuals, 20 of the subjects who in the vaccine arm got infected, but 80 in the meningococcal arm, which would show 80% efficacy. You could also see 30% in protection, which would call it an average vaccine. And if there's no difference between the number of infected people in the COVID arm versus the meningococcal vaccine arm, the vaccine would fail. However, there's one more condition. The vaccine arm could have more people infected than the control arm. And this has happened in the past. And this would give you an efficacy of minus 20%, which means the vaccine could make it dangerous. So it is not obvious which of these results are going to come out. Time will tell what will be expected. It is very important to monitor the performance of the vaccine on a day-to-day basis. And there is usually what is called a Data Safety Monitoring Board, DSMB, which is an unblinded panel of experts who review the clinical trial progress, and they inform the investigators to ensure that nothing unsafe is happening with the vaccine. So these are the possible results that could happen. So the criteria for a successful vaccine are naturally the efficacy of the vaccine, which is the protection from infection. Safety, it should have no or minor side effects, which are all the scientific aspects of the successful vaccine. And the economic, social, and political aspects of the vaccine include value and access, which is how much investment will be required for doing research, clinical trials, and large-scale manufacturing. What will be the price of this vaccine once it is available? How will it be distributed? How will the testing and reporting of the vaccine effects be reported through pharmacovigilance? And then ultimately evaluating the impact of this vaccine. I think the good news is that a lot of philanthropic organizations are collaborating with industry, academia, governments to make sure that this, whatever vaccine is developed is available for as many people as possible. So finally, I'll end with by just showing me a cartoon of what vaccination will do to our population. So once the vaccine is available and sufficient number of people are vaccinated, and the number from experts seems to be somewhere about 60 to 70% of the population should be vaccinated to achieve what is called herd immunity, you will have uninfected, unvaccinated people shown here in green. You will have previously infected individuals who have cleared the virus and are now healthy. They are the black dots. There will be currently infected people who are the red dots, and there'll be very sick individuals who are in the hospital and sequestered. And finally, of course, the vaccinated individuals shown here in blue. What herd immunity enables is that when there's a person that is infected, shown here in red, comes close to a person who's not infected and not vaccinated, shown here in green, there's almost 100% likelihood that this red will convert this green into the red, so infection. However, once these two people are infected, they cannot infect anyone in their circle because either they are vaccinated, which is blue, or they have been previously infected. So then the herd immunity prevents the virus from circulating outside this circle, and hopefully the virus dies out over here. So this is the concept of herd immunity, and we'll see what it takes to develop this. And so finally, I'll end by just saying that I presented a little bit of a summary of the basic immunology. We looked at the components of the virus that may be responsible for inducing protective immunity, what the immune response in the patients would be, a design of a vaccine, and then finally herd immunity. So thank you very much, and I'll be happy to answer any questions.
Thank you, Narendra. What a fascinating talk. Even a non-biologist like me could understand pretty much everything you spoke about. So thank you for that. I'll appreciate if you could stop sharing of the screen so we can see a more smiling Narendra Chirmule. Thank you so much. I think before we get into questions from the audience, and please keep on asking questions, audience. We'd love to hear from you and love you to ask questions from Narendra. I have two questions that my friends have asked me to ask you. So when does the vaccine come?
It's a very good question. I believe that due to the unprecedented collaboration that is happening across the world, we might have a vaccine if everything is successful as soon as the fall of this year. And the reason being that the companies who are developing the vaccines have done a lot of work prior to this. It's not like they just started working on this vaccine just now. I gave you the example of the Chadox vaccine. The Oxford team has been developing chimp adenovirus vaccines for a while. It didn't take them long to plug in that piece of the COVID spike into the already existing process that they have and develop the process a little bit faster. And I think their collaboration with multiple institutions that is in the news, like for example in India, they have tied up with Serum Institute, which is one of the world's largest manufacturing units, will enable this vaccine to be very quick. So I believe, it'll come by the fall of this year. We'll at least have the start of the vaccine. I mean, the large scale manufacturing and lots of people will take at least a year or two.
So a lot of people, Naren, are saying that this vaccine has started mutating. There are different versions coming up. There are rumors about Vietnam getting another mutant. Are these rumors really happening? And if yes, then truly will a vaccine be successful?
Viruses evolve to survive. So when just like humans do, so viruses, when they go through an immune attack, they will try to survive by their only way to escape the immune system is to mutate. And if they mutate and they can overcome the immune response, then they will survive longer. So one thing that I note from the mutations that we see is that the mutations are still relevant to the entry process that coronavirus uses, which is the spike protein entering ACE2 and TMPP RS2 mechanisms. I think if that is the entry mechanism and there are other mutations, then I think if the vaccine protects against the primary mechanism of entry, it should work against the few mutations that we see. One important thing about the mutations that we are seeing right now, the number of mutations that we see with this virus is far, far lower than another, let's say like HIV, for example. So although time will tell what the mutations are, but right now I'm not very concerned about the number of mutations that we are seeing, because I think the vaccine should protect against all of them.
Tell me one thing. This is a very stupid question, and don't laugh at me. They say that you get immunity once you have the COVID virus. And I've also read that it's similar to, let's say, common flu. So why don't I get immunity against common flu, and why will I get immunity against COVID?
Not a stupid question at all, Atul. Really, really insightful question. Let's see if I can answer the question. It's a tough question to answer. The flu vaccine, in 100 years of trying to develop a vaccine, what is called a universal flu vaccine, which protects against all strains of flu, have been disastrously unsuccessful. We haven't been able to develop a universal flu vaccine for a long time. So the flu vaccine process is to make a vaccine every year, and every year the WHO predicts what strain of flu is going to come, and vaccine manufacturers scramble to make the vaccine for that particular year. That's the flu process. Coronavirus comes from a different family of viruses. It's not the same family. And so I believe that the mutation rate of coronavirus is slightly lower. This is speculation. And therefore, the entry process by which we've identified the one I showed you, the bind, cleave, enter process, is quite conserved between the different sequences that we see. And if we can develop an antibody which can block that entry process, the mutations and the different variants will all be blocked. I think it'll be over time we should be able to tell whether coronavirus mutations result in escaping the immune response every season, just like the flu, or whether we can stop it at its tracks. So time will tell. So not an easy question to answer, but that's sort of the high hand
So you're
waving
saying
answer.
that if we are lucky, there'll be a vaccine for time, and if you're not lucky, we have a problem at hand.
Yes, we'll have a problem at hand. As I showed you of the possible results, all the possibilities are existing. And I just showed you that the possibilities of what will happen is all the 80 vaccines that are being tried will give you those range of results. Some of them will be working really well, and some of them not so well, and some of them may be harmful. So we don't know. I think it is important for us as scientists, as a scientific community, to keep our eyes open as to seeing what those results are showing.
So I've got a question from Adesh Sani over here, Naren. He's asking that, sorry, are you, okay, can I go ahead? He's asking if there's any study that shows how long the corona survivors would be producing antibodies. Is there any study for that?
Yeah, very good question. It's a new virus, so we don't know, and I'm not familiar with any studies. I'm not familiar. There may be studies where antibodies to the earlier coronavirus epidemic are measured. But I think if we can extrapolate from other viruses, other highly replicating viruses, like let's say varicella vaccine, which is the chicken pox. One of the things we've learned with the chickenpox live viral vaccine is that if you get infection with chickenpox, which we all do as children, we have a very long immune response to this chickenpox infection, which lasts 20, 30, 50 years, right? And at some point that vaccine, we know that in some of the individuals, that response, the virus-specific immune response wanes because it manifests into what is called shingles, the disease where the virus starts replicating again. And I've got shingles myself, so I know that it's a very, very painful disease. Now, when you give a vaccine, in principle, the vaccine also should do the same thing. But I think in my own experience with varicella, the vaccine lasts for about 10 years or so. The vaccine-induced immune response lasts around 10 years, after which you might need a booster. A similar example I can give for hepatitis B. So in summary, a virus-induced immune response is very long-lasting. A vaccine may be a decade or so.
Excellent. It's a bit of a technical question from Adesh again, and he's asking to generate live attenuated viral particles at commercial scale, which type of cell lines or animal models are used?
Yeah, this is a standard process. I mean, actually, it's not very difficult to make a large scale of attenuated vaccines. Like I mentioned, measles, mumps, rubella, and other vaccines are all live attenuated vaccines. The more recent ones, for example, is the rotavirus vaccine, which is a live attenuated vaccine. And generally, there are a lot of different cell lines that people use. There are Vero cells, there are 293 cells. These are all commercial cells which have been used to grow large-scale vaccines. In fact, when you have a manufacturing facility, you want a virus which grows nicely and rapidly, because then you're making a lot of product. But the challenge is to purify out the pure virus and ensure that there's no contaminant viral contaminant in the vaccine. So the challenge lies more in the purification rather than the growth.
So let's move a little beyond the vaccine. What about cures of the disease? Any indication of anything coming in the near future?
Well, I'm sure all of us do the same thing. We get up early in the morning and look at the news, open our phones and say, okay, what's the latest? And I was really excited this morning to hear Anthony Fauci announce from the Oval Office that remdesivir has shown a very, very positive result in a very controlled clinical trial. Like he's my hero in terms of public speaking. And I really recommend everybody to listen to that 5 or 10 minute speech. It is going to go down as one of the most landmark speeches. It will be one of those I have a dream kind of speech. So please, it's a wrong take wali speech. But he showed that remdesivir has a positive result, which is very exciting because I think with a lot of speculation going on with which drugs work, which drugs don't, it is nice to see a targeted drug working with the mechanism of action we think it works on. Like remdesivir binds and inactivates the RNA polymerase. And when given to patients who are sick, you can actually see clinical effects of that. So it's a controlled study. There are many other drugs. I think we undermine the drugs that we use. For example, plant-based drugs, or that Shulini, specializes in. I think there are a lot of opportunities to make very, very effective drugs, not just for the viral application, but also for overall immunity and development. So I think the role of holistic medicine is highly underappreciated. And I think there'll be a lot of opportunities to help understand immune responses using holistic drugs.
Manav is asking, when can we expect to live the way we used to live before Corona? I know that's nothing to do with the talk today, but of course, if we get a vaccine, it'll be back to normal.
Philosophically, personally, I hope we never go back to the way we lived before. I hope we don't pollute the environment. I hope we, don't climb over ourselves, politically and socially, and I hope we work together like we are working now. I hope we start education like the way we are doing now, right, Atul?
Absolutely. So that we promise you, Naren. Wherever you are in the world, we're not going to leave you. Even if you go to the moon and settle there, we're going to beam you over to Shulini every week.
I'll be there. Thank you. No, but I personally, I feel I don't want to go back to the way we lived. I feel. But the reality is that it will come through stages, and I think each country will have to make its own decisions of how to bring the economy back. And I'm not trivializing how difficult it is going to be for a lot of people, and how difficult it has been for a lot of people. I think everybody has to play some role to come back to normal. So it's really hard to speculate.
There are lots and lots of questions here, and I'm going to pick up 2, 3. But Naren, if you could just type your email address or the chat bot, people I hope can reach out directly to you if they have any specific questions. But I'll take 3 more. Time is short. Anuradha is asking, will the vaccine have differential effect based on our genotype? Maybe works in the US, but not in Asians.
No, yeah, okay, good question. It will definitely have a differential effect in different individuals, that's for sure. It won't be different by race, it won't be different by countries, it won't be different in that manner. I mentioned very briefly about the MHC molecule, the major histocompatibility molecule. This is the molecule that we inherit from our parents, one from each parent. And that's why we are restricted to the MHC sequence, which is present only in us. And the MHC molecule, like I showed you in that cartoon, is responsible for presenting the antigen to the immune system. So each one of us has a different MHC, and each one of us presents the antigen in a slightly different way. And therefore, the strength of the immune response that each one has is slightly different. And that's why you will see a bell-shaped distribution of the immune response, where some people may not respond at all, and some people may respond extremely vigorously. But most of us will be responding in that bell-shaped curve. So I think the variability of individuals is what keeps the species alive. If we all responded exactly the same, on the flip side, we all would have been eliminated by the same virus.
Wow. One question by Dr. Rosie. She's asking, can there be a new novel coronavirus from an animal source that can affect humans again?
Never say never, right? I mean, I think the way humans, we are progressing along our evolutionary path, there will be new viruses and vectors and combinations. I think what we learned from this epidemic or pandemic is unprecedented. I think the global focus on developing processes to to make sure that something like this doesn't happen, or if it happens, we have a process to manage it, will be important. I don't think we can ever prevent these kind of combination of things to happen again. So let's hope, we have things under control in the future.
So lots of questions, Naren, coming about people who have recovered from corona and they're getting infected again. There's some rumors again from China. What's your, again, sense on that? Do you think someone who's infected by corona, will he or she get infected again, at least in the near term? Let's say the near term is 6 months, or that still we don't know about it?
Yeah, very good question. I think this is my understanding of the science of what's happening with the coronavirus that is circulating today. I think the mutation rate is not as high today as I see it, where a new strain will come and start infecting people all over again in this very short term. It might, but I don't see that happening, at least the way I see it. So the reinfections that are happening, I assume very small in number, and they could be for several reasons. One could be that the initial test that they did, or the initial symptoms that they had, it came up negative, and now it's coming up positive. So it looks like a reinfection, which it may not be. The second could be that, in fact, there is a mutation and that people are getting reinfected by a mutation, but I think that possibility is low. And the third could be, that the mutations, that the virus was not eliminated completely by the immune system. There was some reminiscent virus that remained, and the immune system got rid of the first wave of the virus, so to speak, and the second wave started coming up again. So those are the three options of why reactivation could occur. But I don't think, at least my personal opinion, based on the things that I'm seeing now, that reinfection will be a major issue in the short term. But it's all about the data. I haven't seen the actual data from the reinfected people.
Okay, two last questions, two last thoughts. First, from me, looks like a lot of optimism from your end. Naren, you're saying there's a drug which is out there. Today they announced. Secondly, a vaccine could be coming by the fall. So should I go and buy shares now? Can I invest in the stock market?
I think the risk is yours to take, but I'm feeling pretty optimistic. I'm feeling very optimistic about things happening. The scientific, social, economic, and all these communities have come together in ways that I cannot imagine. And someone like you, who's following all aspects of these, in your fantastic talks that you've been coordinating, and you're getting knowledge. So you're getting so much knowledge and diverse information. I think it's hard to be not optimistic. And what choice do we have? We have to be optimistic. I think it's really a fascinating time that we are living in.
Absolutely, we're learning how to relive again. I'm actually enjoying it. I'm enjoying these sessions. Of course, I hope and pray that we are over Corona very, very soon. The economics gets better. But more importantly, we don't go back to the crazy way we lived as human beings. Just one last question, which I think is very pertinent. I forgot the name of the person, but someone asked this question saying, you are such a wonderful, or Vend actually is asking, you're such a wonderful speaker. Who's your public speaking hero?
There are many public speaking heroes that I have. I never, I don't know if I'm a good speaker or not. I think it's for you to decide. But you used to be stammering and talk a lot of different things. And I'll tell you a thing, I attribute this to my music teacher, Tara Kinney. For the last 4 years, especially for the last 4 or 5 years now, I've been learning vocal music. And I would recommend everybody to go and learn vocal music, even if you have a bad voice, you think, right? Because what happens with vocal music is you are forced to sing. And when you sing, your voice becomes strong, your vocal cords become strong. And you're able to modulate your voice very nicely because you have a good voice. And when you can do that, you can impress people. So I would say, in order to get better, listen to TED Talks, listen to good people, and go and learn singing is my advice for getting good public speaking.
So before we close, I have to go on.
Yeah, no, Anthony Fauci, I'd mentioned Anthony Fauci in the beginning. He's sort of my role model, one of my many role models for a good talk.
What do you think about Donald Trump?
No, he's also very good speaker. Actually, he expresses him so well. We may agree or not agree, and we might be on different sides of the coin. But people listen to him. It's almost addicting to listen to him.
Fantastic. Okay, so a lot of people don't know, but Naren plays the flute exceptionally well, and he's a great singer. So I think before we say goodbye, if your flute is nearby, you want to just play a half a minute of your flute?
Yes. Thank you.
So this, in the mountains, in the Himalayas, they say that music, especially from the flute, kills away evil, takes evil away. So hopefully the small performance of yours will kill the coronavirus. We hope that. We also, in the spirit of music, I'd like to announce to everyone that we're very lucky at 9 o'clock India time today. We also have the very famous, the Badshah of music, Shafqat Amanat Ali Khan with us, talking to us live only for Shulani at the Yogananda Guru series. So please join us on YouTube at the Shulani University channel, 9 p.m. today. Once again, Naren, thank you very much. A fascinating talk. We'll keep talking to you. You've also promised that you're going to speak to school students and teachers, and I am going to pick up that offer. But I'm sure everyone is waiting to hear back from you. Once you get more news about the virus, we'd love to hear more what's really happening. I hope all of you enjoy this absolutely fascinating discussion. We'll keep coming back to you on Expert Talks. Till then, goodbye. Have a wonderful day and wish you good luck. For people who don't know, Naren is in the US right now. It's very early for him, and he's been very kind to speak to us. Thank you, and have a safe day, great day, and may it continue to rain. Bye-bye.
Thank you.